Hi, this is Paul Thomas, Senior Editor with Pharmaceutical Manufacturing at Interphex 2010, talking with Jim Akers of Akers Kennedy and Associates, aseptic processing experts. Jim, thanks for being with us today.
Thank you.
Appreciate it.
I appreciate the opportunity. Thanks.
Jim, one of the things that we were just talking about is quality by design and applying quality by design principles to aseptic processing. What are some basic thoughts that you have?
Well, I think the most fundamental thought I have is that if we're going to have a quality design in aseptic processing, because the human plays such a critical role as a source of contamination and therefore risk, in order to effectively perform quality by design, that should be, in my humble opinion, the primary consideration. And obviously there are two things we can do. We can reduce human-borne contamination, or we can eliminate it. And as you and I have discussed in the past, fortunately there are technologies moving along that and that are being implemented that enable us to effectively eliminate human-borne contamination in ways that wouldn't have been possible a few years ago.
But for most manufacturers, it's not reasonable to go directly to that new scenario that you're talking about, completely eliminating the human element. How can they transition in that direction? How can their QBD efforts help them to transition towards that eventual scenario?
What I like to say, and a couple of my colleagues say frequently, is that there's no such thing as a good intervention. There's no such thing as a risk-free intervention in aseptic processing. So the first and most sensible thing that can be done is to review your processes extremely critically and determine to what extent you can minimize human impact on those processes. You know, to the extent that you can eliminate interventions, you will have built quality into your system in a manner that it may not have existed before. I think as an industry, we've tended to be rather intervention tolerant, believing that as long as we have people well gowned and we have good airflow and this sort of thing, that will take care of the risk associated with human intervention. Experience has taught us that while those are factors that can reduce the impact of human interventions, they don't eliminate it. So the best intervention is one that doesn't occur. And we've had some success going through and looking at aseptic processes and helping people come to grips with the number of interventions they have and see that if we can eliminate them, and in many cases we can.
And you said some successes, but maybe not enough successes, and you've talked a lot about the food and beverage industry, how that industry is in many ways more advanced in terms of automation, in terms of aseptic practices. Right. Why is that, and what can Pharma do to start moving towards those things that are already implemented in food and beverage?
Well, I think the food and beverage industry has come from a different culture and has accepted that the only way to really do aseptic processing is with full separation of the human operator from the environment. And I don't think Pharma has quite accepted that. I think we've taken in our industry more of an approach that if we train people well enough and use good enough gowns and do a lot of monitoring and testing, that we can obviate some of the risks associated with personnel, or at least understand them. And I think the food and beverage people have it closer to right. We started off talking about quality by design, and I would offer that quality by design is possible. Quality by test is not really possible. We can't test quality into an aseptically controlled product. So we reach a point where we can do more personnel monitoring, we can do more environmental monitoring, but it is not, for a number of good analytical reasons, going to shed much useful light on how well our processes are controlled. To do that, we have to go back and really design the process properly, and that means become intervention intolerant to the extent that our systems allow us to do so.
But you talk about quality by design, but the food and beverage industry hasn't necessarily had an overarching initiative such as QBD, so how has it managed to move forward?
I think the commercial motivations. They found that by using very advanced aseptic technologies, they can extend shelf life of their products, which manifests itself commercially. They can reduce spoilage. They can reduce customer concerns. So they are, interestingly enough, I believe, more purely commercially motivated than they are regulatory motivated. And interestingly enough, they've ended up in a bit different place. So I think that the culture of practice, as a result of that, on their side of the aseptic processing technology, has evolved differently than it has in pharma.
And my last question will be regarding the current economic downturn. Has that actually stifled some of the innovation that you would hope to see in the industry, or are you seeing maybe manufacturers taking this opportunity as a way to try and take a big leap forward and implement some new technologies?
Well, it's a complicated question with kind of a multiphasic answer, and I think we are experiencing a slowdown, and certainly there's been a lot of projects put on hold in the pharma industry, but I do think that this pause is giving people an opportunity, which they are taking, to reconsider how they might move forward and to have a harder look at newer technologies. And I think at the same time, we have coming along new categories of products that are largely biologically derived, regenerative medicine, cytotherapy products. And those products, even though they're quite different from the aseptic beverage circumstance, in that those are very high throughput facilities, and the cytotherapy and regenerative medicines are not. They're very low throughput. But nevertheless, those people are availing themselves of the idea of automation and separative technologies to reduce the very profound contamination risk associated with their products. And because their products are custom, we really can't test quality into them. They're delivered to the patient before they would come off sterility test. So that is creating an opportunity, I think, to implement these more advanced technologies with very high value products that require special manufacturing circumstances. So I see it coming along in both of those facets.
There is a silver lining then.
Yes, very much so.
Good. Jim, thanks so much for joining us today. Really appreciate it.
Thanks, Paul. Thanks for the opportunity.
Thanks.
Thank you.